Welcome, curious minds, to another deep dive. Today, we're plunging into a scientific mystery that, well, for over 50 years, it completely baffled the brightest minds on the planet.
Yeah, it really did.
I'm talking about a grand challenge in biology, a problem that felt almost insurmountable. Now, imagine an artificial intelligence program not just making headway, but essentially solving it. And in doing so, fundamentally reshaping our understanding of life itself. That's the astounding story we're here to unravel today.
It really is one of those rare, truly paradigm-shifting moments in science. What was once considered this painstaking, often impossible task
Right.
has been transformed almost overnight, it feels like, into something achievable and crucially widely accessible.
You're right. It's incredible.
Yeah.
We're taking a deep dive into AlphaFold. That's the AI program developed by DeepMind, which is part of Google now.
Right.
This program has achieved something that scientists used to just dream about. It predicts the precise three-dimensional shapes of proteins, and with an accuracy that was once thought to be completely out of reach without, incredibly laborious and expensive lab experiments.
And this isn't just some technical footnote in AI development. It's a breakthrough that's been widely heralded as transformational by the scientific community.
Absolutely.
I mean, its creators, Demis Hassabis and John Jumper, they were even recognized with a portion of the 2024 Nobel Prize in Chemistry, specifically for protein structure prediction.
Wow.
That's a rapid and profound ascent for a computational tool. It really signifies its massive impact.
It really is an astonishing achievement. So for you, our listener, our mission for this deep dive is to truly unpack what this protein folding problem actually entailed.
Yeah, what was the big deal?
Exactly. We'll explore how AlphaFold's unique AI approach managed to crack it, the immediate and profound impact it's already had on science and medicine, what exciting future advancements are on the horizon, and even some of the deeper ethical and philosophical questions this kind of AI-driven discovery raises for all of us.
Right, the bigger picture stuff.
I think you're going to find some real eye-opening moments as we go through this.
I think so, too.
So let's start at the very beginning. Let's set the stage for this decades-old protein folding problem. To truly grasp AlphaFold's achievement, we first need to understand the stars of the show. Proteins.
Right.
What are they, and why is their shape so incredibly, fundamentally important?
Proteins are, well, they're truly the fundamental building blocks and really the workhorses of every living thing, from bacteria all the way up to us.
Workhorses, I like that.
Yeah, you can think of them as long, linear chains, much like a string of beads, and each bead is a smaller unit called an amino acid.
Okay.
But here's the crucial insight. These chains don't just stay linear, they don't just float around like floppy strings. They reliably, almost magically it seems, fold themselves into highly specific, intricate, three-dimensional shapes.
So the shape isn't just like a random outcome. It's absolutely fundamental to what these proteins actually do.
That's exactly right. Imagine a protein as maybe a specialized tool. Its unique 3D shape is the working part. It dictates everything.
Everything.
Pretty much. If it's an enzyme, its precise shape creates what's called an active site, and that allows it to drive specific metabolic reactions in your body.
Ah, okay.
If it's an antibody, its shape lets it precisely bind to and fight off an infection. And if it's a structural component, like collagen, its shape gives cells their rigidity or flexibility.
So it's like a specific key needing to fit perfectly into a specific lock for a function to happen.
That's a perfect analogy. Without that exact shape, the tool is useless. The key simply won't turn the lock. The protein just doesn't work.
Okay, so if that perfect shape is everything, then predicting that shape just from its basic amino acid sequence,
Yeah,
that must have been the ultimate quest.
absolutely.
And that, I gather, is the essence of the protein folding problem. It sounds incredibly difficult.
Incredibly is almost an understatement. It was considered, monumentally difficult. And for a very good reason. Theoretically, a chain of amino acids, let's say just a few hundred links long, which isn't even a particularly large protein,
Right.
it could potentially fold in an astronomically vast number of ways. I mean, we're talking about more possible configurations than there are atoms in the observable universe.
Wow, it's hard to even comprehend.
It really is. Yet in nature, each unique amino acid sequence reliably, almost instantly, folds into the correct functional shape every single time.
Okay.
Replicating that predictive power computationally was the grand challenge. And for decades, honestly, it seemed like an impossible dream for many scientists.
So before AlphaFold came along, How did scientists even try to tackle this? I mean, they weren't just sitting around waiting for AI.
No, certainly not.
Hmm.
Traditionally, determining a protein structure required immensely laborious, time-consuming, and very expensive laboratory experiments.
Like what kind of experiments?
Well, the gold standards were techniques like X-ray crystallography, or more recently, cryo-electron microscopy, or cryo-EM for short.
Okay, I've heard of those.
Yeah, but these methods demand specialized equipment, highly purified proteins, which can be hard to get, and a great deal of expert, hands-on work. It often takes months, sometimes even years, for a single protein structure.
Months or years for one structure. Wow.
Yeah, it's a bit like taking an incredibly complex, high-resolution photograph of something microscopic and totally invisible to the naked eye. Very painstaking.
And computationally. What progress did we see on the software side before AlphaFold?
On the computational front, progress was, well, let's say incremental, over decades. By around 2016, for the really difficult targets, the best software could only achieve moderately accurate structures.
How moderate are we talking?
To give you a benchmark, they used a quality scale called the Global Distance Test, or GDT. Scores are out of 100. The best methods were scoring around 40, maybe 50 on a good day for hard targets.
40 out of 100, that sounds not great.
It meant a significant error margin, yeah. The predicted shape was often quite different from the real functional one. Many researchers also relied heavily on something called homology modeling.
Homology modeling. What's that?
It's basically trying to find a known structure of a similar protein, one that's already been solved experimentally,
Okay.
and using that known structure as a template to guess the structure of your unknown protein.
So like using the blueprint of a Ford Model T to guess the design of a modern car.
Slight chuckle. Sort of, yeah. It works okay if the proteins are very closely related. But as you can imagine, this method completely failed if there was no similar protein structure already known.
Right. So if you were trying to predict the structure of a completely novel protein, maybe from a newly discovered organism or one involved in a rare disease.
You were simply out of luck with homology modeling. Your analogy was spot on trying to build a new rocket with only early airplane blueprints. If it's truly novel, you're lost.
Okay. And just to really appreciate the task AlphaFold faced, can you quickly walk us through the folding process itself? You mentioned primary structure.
Right, so you start with that linear amino acid chain, that string of deeds. That's the primary structure.
Got it.
This chain then starts to fold into local repeating patterns, like little coils called alpha helices or flat sheets called beta sheets. That's the secondary structure.
Okay, coils and sheets.
Then these secondary structures compact and interact with each other, folding up into a unique overall 3D shape. That compact shape is the tertiary structure that's the functional form for most proteins.
And sometimes it gets even more complex.
Sometimes, yes. Multiple folded protein chains, multiple tertiary structures, can assemble together into an even larger complex. That's called the quaternary structure, like hemoglobin in your blood.
Okay, so AlphaFold's monumental task was to predict that final correct 3D shape, the tertiary or sometimes quaternary structure.
From only the initial linear amino acid sequence, the primary structure, nothing else.
Wow. Like guessing the final intricate origami sculpture just by knowing the sequence of folds written on a piece of paper?
That's a good way to think about it, yeah. But with millions, billions, trillions of possible wrong ways to fold it.
All right, so we've established this massive challenge, finding a needle in, as you said, an astronomical haystack, or maybe even a galaxy of haystacks.
Chuckles. Yeah, something like that.
How on earth did AlphaFold even begin to tackle something so seemingly impossible? What was so revolutionary about its AI approach?
Well, AlphaFold, developed by DeepMind, took a fundamentally different path. It employed deep learning, but on a massive scale. Instead of trying to explicitly program all the incredibly complex rules of physics and chemistry that govern how a protein folds.
Which scientists have been trying to do for decades, right?
Exactly, with limited success. Instead, AlphaFold effectively learned these rules directly from data. It was trained on an enormous data set, over 170,000 known protein structures that had been painstakingly determined experimentally over those decades.
Okay, the ones from the protein data bank.
Right, from the PDB. And alongside those structures, it was fed millions and millions of related protein sequences from various organisms.
So it's less like DeepMind coded, okay, AI, if you see amino acid X next to Y, then fold left and consider the van der Waals forces.
Right. Definitely not like that.
It was more like, here are 170,000 finished, incredibly complex buildings, plus millions of sketches of similar buildings. You, AI figure out the underlying architectural principles yourself.
That's a fantastic analogy. It's like giving the AI access to every architectural blueprint ever drawn, plus photos of the finished structures, allowing it to learn the deep principles of construction, how different materials connect, how structures achieve stability, how form relates to function, rather than trying to program those rules explicitly case by case.
That is a crucial distinction. So how does this AI actually think then? What's happening inside its neural networks that allows it to figure this out?
Well, the system uses a pretty sophisticated neural network architecture. One of the key components, especially in the later versions like AlphaFold 2, is something called an attention mechanism.
Attention mechanism. What does that do?
It essentially allows the AI to effectively focus on different parts of the protein sequence simultaneously. And crucially, it helps it understand the critical relationships between pairs of amino acids, even if they're really far apart in that linear sequence.
Ah, okay. So it's not just looking at the bead right next door. It's looking at how a bead way down the string might interact with one near the beginning,
Yeah.
seeing the whole potential sculpture at once.
Precisely. It's considering the global context. In essence, the AI learns these incredibly subtle biochemical and geometrical patterns, rules, if you like, about how different segments of a protein tend to come together in 3D space.
And it doesn't just look at one sequence, right? You mentioned millions of sequences.
Right, and that's another key ingredient. It cleverly leverages evolutionary information. If you look at many similar protein sequences from different organisms, say, the sequence for hemoglobin in humans, chimps, dogs, fish, you gain vital clues. If two specific amino acid positions consistently change together or co-evolve
Yeah.
across different species, it's a very strong indicator that they are likely physically close to each other, interacting within the folded structure.
Why is that?
Because a mutation in one position might destabilize the structure, but if a corresponding mutation occurs in the position it interacts with, it can compensate and restore stability or function. So tracking these co-evolving pairs gives powerful hints about 3D proximity.
Okay, that makes sense. So it's using evolution as a guide to spatial relationships. And this all culminated in AlphaFold 2 in 2020, which you said was described as a radical departure. What made that version the real game changer?
AlphaFold 2 was indeed revolutionary because it produced what's called an end-to-end prediction of the protein's shape.
End to end.
Yeah, think of it like this. Older methods often involved multiple separate steps or programs. Maybe one predicts distances between amino acids, another tries to build a model based on those distances, another refines it using physics, like a kind of pipeline.
Right, like an assembly line.
Exactly. AlphaFold 2 was different. It was more integrated. It iteratively refined its own predictions. It would generate an initial 3D model, then feed that predicted structure back into its own neural network as additional input, allowing it to reassess and improve the structure in subsequent cycles.
Ah, so it learns from its own attempts. It recycles its work.
Precisely. It keeps refining its output, almost like sculpting, until it converges on a high-confidence model, and then it outputs the final 3D coordinates for every atom, along with confidence estimates for each part of the structure. This tells scientists how reliable it thinks each region of its prediction is. This iterative, end-to-end self-refining process was fundamentally different and much more powerful than earlier approaches.
I remember reading that David Baker, who's a real giant in protein science, remarked it was an amazing achievement, like what they did with Go,
Yeah.
comparing it to AlphaGo, the AI that beat the world's best human Go masters.
Hmm.
That really puts it in perspective. It suggests a similar leap, maybe, in AI capability.
I think that comparison is very apt because it signifies one of those moments where AI moves beyond just being a very powerful calculator. It seems to achieve a level of intuition or pattern recognition or problem solving that approaches or even surpasses what humans could do even after decades of focused effort on that specific problem. It felt like a cognitive leap.
So why is this approach so effective? What makes AlphaFold a genuine paradigm shift
Yeah.
compared to everything that came before? Let's really break down the practical differences.
What's core innovation was that holistic approach combining the sequence information, the deep evolutionary data, and its learned understanding of biochemical and geometrical constraints, all within this sophisticated deep learning framework. It modeled the whole folding process much more comprehensively, and this is where it truly just blew traditional methods out of the water.
Okay, let's compare them directly. First, big difference. You mentioned older methods often needed a template, a similar known structure.
Exactly. That was a huge limitation. Older computational methods, especially homology modeling, were heavily reliant on having a known structure of a similar protein to use as a starting point, a template.
Oh.
AlphaFold. It needs no template.
None at all.
Essentially, no. It excelled at the CASP competition targets, where no similar structure existed in the database. These were called free modeling targets, and they were previously considered almost impossible to predict accurately using software alone. AlphaFold handled them remarkably well. That's a massive hurdle cleared.
Okay, no template needed. What about accuracy? You mentioned that dismal 40 or 50 out of 100 GDT score earlier for the hard targets. How did AlphaFold compare?
The change in accuracy is frankly jaw-dropping. AlphaFold achieves accuracy comparable to experimental methods for a large fraction of proteins.
Comparable to lab experiments,
Yes.
seriously.
Yes. In that pivotal 2020 CASP-14 challenge, AlphaFold scored above 90 out of 100 on that GDT accuracy scale for about 2/3 of the target proteins. Remember, older methods were struggling around 40-50 on difficult cases. A score above 90 GDT means the predicted structure is generally considered to be highly accurate, often virtually indistinguishable from the structure determined experimentally in the lab.
That's unbelievable. So years of lab work versus how long does AlphaFold take?
That's the other incredible part. Lab work takes months, often years.
Yeah.
AlphaFold is not only accurate, it's incredibly fast. A typical protein structure can be predicted in days, sometimes even hours or minutes, depending on the protein size and the computational resources.
Days or hours versus months or years.
Exactly. This allows for immense throughput. And consider this. By mid-2022, the AlphaFold database contained predictions for over 200 million protein structures. Compare that to only about 170,000 unique experimental structures deposited in the PDB over 5 decades.
Wow. That completely redefines the scale of structural biology. It's like going from hand copying a few manuscripts a year to having a digital library of almost every book ever written.
That's a really good way to put it. The sheer volume of structural information suddenly available is staggering.
And you mentioned accessibility earlier. Is this power locked away in Google's labs, or can anyone use it?
That's another critical point and a huge win for the scientific community. DeepMind made a landmark decision. They made AlphaFold's source code open source.
So any lab can run it.
Yes, if they have the computational resources. And perhaps even more impactfully, they collaborated with EMBL-ED to create the AlphaFold protein structure database, making all those millions of predicted structures freely available online for anyone to download and use.
That's huge.
It effectively democratizes access to cutting-edge structural knowledge. A scientist anywhere in the world, even in a low-resource setting without expensive crystallography gear or cryo-EM microscopes, can now look up the likely structure of their protein of interest. It's a powerful equalizer in research.
Okay, so just to summarize what makes AlphaFold so unique and revolutionary. It learned the hidden rules of folding directly from vast amounts of data, rather than being explicitly programmed with human-derived rules.
Nice.
It captured subtle biochemical and geometrical patterns that were beyond previous algorithms. It needs no template, achieves near experimental accuracy for many proteins, works incredibly fast, and its results are freely available to everyone.
That's a perfect summary. It effectively, as some have said, turned a problem once thought to require decades of work into something solvable in minutes. That's the paradigm shift. It shows the profound power of AI when applied thoughtfully to fundamental scientific questions. It's not just a faster way to do the same thing, it's enabling a completely different way to understand biology.
Okay, so with this truly revolutionary AI approach shaking up the field, what has been the immediate and profound impact of AlphaFold? Where are we already seeing these key scientific and medical achievements playing out?
Well, its arrival was really announced with a bang at the CASP competitions. CASP stands for Critical Assessment of Protein Structure Prediction, and it's basically the Olympics for this field.
Right, where different teams test their prediction methods.
Exactly. AlphaFold first made major headlines by ranking #1 in the prestigious CASP-13 competition back in 2018. It significantly outperformed other methods, especially on those really hard, free modeling targets, the ones without known templates.
So that was the first big signal that something new was happening.
It was, but then AlphaFold 2 came along for CSP-14 in 2020, and that just completely changed everything. Its performance was a true shockwave.
How much better was it?
It achieved such unprecedented accuracy across the board that many influential figures in the field declared it had essentially solved the long-standing protein structure prediction problem, at least in terms of predicting the static structure of single protein chains.
Solved is a strong word in science.
It is. John Malt, one of the CASP organizers, stated quite plainly that AlphaFold 2 had largely solved the problem and had shifted protein science forever. Another prominent structural biologist, Mohammed Al-Quraishi, recalled being in shock when he saw the 2020 results, saying many people in the field had not expected to see that level of progress within their lifetimes.
Wow. Imagine dedicating your career to chipping away at a problem, and then suddenly an AI comes along and just leaps over the finish line.
Yeah, the reaction was a mix of amazement, perhaps some disbelief, and ultimately huge excitement.
And rather than keeping this incredibly powerful, potentially very valuable tool proprietary, DeepMind made a different choice, didn't they? You mentioned open science.
Indeed, and this is arguably one of its most commendable achievements beyond the technical prowess. In July 2021, DeepMind published the detailed AlphaFold 2 methodology in the leading scientific journal Nature. And importantly, they made the source code openly available to everyone.
So anyone could see how it worked and even try to build upon it.
Exactly. And simultaneously, they launched the AlphaFold protein structure database in partnership with Europe's IMBL-EBI, the European Bioinformatics Institute.
This database sounds like a monumental undertaking.
Yeah.
What did it actually contain at launch?
At launch, it was already massive. It released predicted structures for nearly the entire human proteome. That's the complete set of proteins expressed by our bodies, tens of thousands of them.
The whole human set. Wow.
Pretty much. And also the proteomes of 20 other critical model organisms used widely in research like yeast, E. coli, fruit flies, mice. This amounted to over 365,000 protein structures made freely accessible right from day one.
That's incredible, and it grew from there.
It grew exponentially. By mid-2022, just a year later, this database had expanded to include predictions for around 200 million proteins from practically all known sequenced organisms. Basically, as they put it, structures for nearly every known protein on the planet.
200 million compared to the 170,000 experimental ones. That's just mind-boggling scale.
It is, and this enormous, freely accessible data set has been accessed, they report, by over 1 million researchers in more than 190 countries. It's been described again and again as a powerful democratizing advance because it puts this cutting-edge structural information into the hands of any scientist, anywhere, regardless of their institution's wealth or experimental resources.
That's a huge step towards making science more collaborative and, frankly, more equitable. How has this flood of new structural information actually accelerated research within structural biology itself? Are the experimentalists using it?
Oh, absolutely. Structural biologists now routinely use AlphaFold predictions to aid their experimental work. It hasn't replaced experiments, but it's become a powerful partner.
How so?
Well, for example, in X-ray crystallography, determining the structure often involves a difficult step called phasing, or using molecular replacement, where you need a good starting model to interpret the experimental data. AlphaFold models are now frequently used as incredibly accurate starting templates for this.
So it gives them a massive head start.
Exactly. It has even solved structures where all previous attempts using experimentally derived templates had failed, simply because the AlphaFold model provided such a good initial guess. It bootstraps the experimental process.
So instead of spending months or years just trying to get that first rough model, they can often jump right to refining a high-quality prediction against their data. That must save immense amounts of time and effort.
Precisely. Difficult structures that once took years of painstaking work to determine can sometimes be solved experimentally in mere hours or days now, just by fitting AlphaFold's model into the experimental data from crystallography or cryo-EM.
Can you give an example of that?
A really striking example is the resolution of the structure of the nuclear pore complex. This is a truly massive molecular machine made of about 1,000 different protein subunits that controls everything going in and out of the cell nucleus. It's like the gatekeeper of the genome.
1,000 proteins. That sounds incredibly complex.
It is. Determining its structure was a huge challenge, but researchers were able to use AlphaFold to predict the structures of many of the individual component proteins and then fit these predicted models into lower resolution cryo-EM maps of the entire complex, like assembling a giant, incredibly intricate 3D jigsaw puzzle. This gave us the first near-complete structural view of this vital cellular machine.
Wow, so it enabled tackling complexity that was previously out of reach?
Exactly. It has become an indispensable assistant, dramatically speeding up structure determination and allowing scientists to tackle much larger and more complex biological systems than before.
Okay, so it's accelerating basic structural biology. But how is AlphaFold impacting our understanding of diseases and, maybe more importantly for many people, helping us find new ways to treat them?
This is where the impact becomes very direct and very hopeful. AlphaFold has already contributed significantly to medical research by revealing the structures of numerous disease-related proteins, many of which had resisted experimental determination for years.
Like what kind of diseases?
A very timely example was during the COVID-19 pandemic. In early 2020, before experimental structures were available for all the viral proteins, AlphaFold was used to predict the structures of several key SARS-CoV-2 proteins.
Really? That early?
Yes. For instance, they predicted the structure of a viral protein called ORF3A. Later, when the structure was determined experimentally in the lab, AlphaFold's prediction turned out to be remarkably close. This provided crucial early insights into how the virus might function and interact with our immune system, even before the slower experimental work could catch up.
That's incredibly valuable in a fast-moving pandemic situation.
Absolutely. And beyond viruses, it's being used extensively now to study proteins involved in cancer, targets for neurodegenerative diseases like Alzheimer's and Parkinson's, proteins involved in antibiotic resistance, and countless other health-related proteins that were previously just sequences in a database with unknown shapes.
In knowing the 3D structure,
Yeah.
that helps understand the disease.
Immensely. Knowing a protein's 3D structure is like having a detailed molecular blueprint. It illuminates exactly how specific mutations, maybe ones linked to a genetic disease, might disrupt the protein's function, perhaps by altering its shape or blocking an important interaction site. And critically for medicine, it reveals potential places where a drug might bind.
Ah, the drug discovery angle. That seems like a huge potential impact. Tell us more about that.
Indeed, perhaps its most celebrated and potentially transformative impact is opening entirely new avenues for drug discovery. By knowing the precise 3D shapes of disease-causing proteins, researchers can much more effectively identify specific pockets or active sites on the protein surface where a drug molecule could potentially bind and modulate its activity, either blocking it if it's harmful or maybe enhancing it if its loss causes disease.
So it guides the chemists, showing them where to aim.
Exactly. It enables what's called structure-based drug design on a much wider scale. For example, the nonprofit Drugs for Neglected Diseases Initiative, or DNDI, they explicitly called AlphaFold game-changing technology for their work.
Why was it game-changing for them?
Because they work on diseases like Chagas disease and leishmaniasis, which affect millions in the developing world, but often lack the massive funding needed for traditional, slow drug discovery pipelines. AlphaFold provided them with high-quality structural models of parasite proteins almost instantly, helping them identify how potential drug molecules could bind and guiding the development of desperately needed new therapies much more efficiently.
So it's not just benefiting big pharmaceutical companies with deep pockets, but also accelerating work on diseases that affect the world's poorest populations. That's fantastic.
It really is. Researchers working on malaria vaccines have used it to design better antigens. Scientists developing new cancer therapies are using it to understand resistance mechanisms or find new targets. It's having a broad impact.
Any non-medical examples?
Yes. In a really innovative environmental application, scientists used AlphaFold predictions to help them engineer better plastic-eating enzymes. They could predict how specific mutations would affect the enzyme's stability and shape, allowing them to rationally design modified enzymes that could break down plastic waste more efficiently. It's truly a versatile tool for molecular engineering, not just medicine.
It certainly sounds like AlphaFold has earned a lot of praise and made a huge splash. What about formal recognition from the broader scientific community? Did the big awards follow?
Oh, absolutely. The scientific community has overwhelmingly recognized AlphaFold's achievements with numerous major accolades. The main 2021 AlphaFold paper published in Nature became one of the most cited scientific papers in modern biology almost overnight, accumulating tens of thousands of citations in just a couple of years, a truly astonishing rate indicating how widely it was being used and discussed.
Tens of thousands of citations.
Yeah.
That's remarkable.
It really is. Then, in 2023, DeepMind's leaders on the project, Demis Hassabis and John Jumper, received the prestigious Breakthrough Prize in Life Sciences, which comes with a significant monetary award, and also the Lasker Award, which is often seen as a precursor to the Nobel Prize in the U.S.
And then the Nobel itself came calling.
Indeed. As we mentioned right at the start, in 2024, a portion of the Nobel Prize in Chemistry was awarded specifically for protein structure prediction, primarily recognizing the transformational impact of AlphaFold.
Oh.
It's actually quite rare for a purely computational tool or method to earn these top biomedical science honors so quickly. It really underscores how profoundly AlphaFold solved the long-standing challenge of protein folding in a way that few had imagined possible just years before, and how central AI has now become to biological discovery.
So after largely solving the challenge of predicting single protein structures, which is just incredible in itself, what's next? Where does AlphaFold or this field in general go from here? What are the next frontiers and advancements we can expect?
Well, AlphaFold isn't standing still. Having made such a huge impact on single protein structure prediction, the focus is naturally shifting to even more complex biological problems. DeepMind announced AlphaFold 3 in 2024.
AlphaFold 3. Okay, so what's new?
This updated AI system dramatically goes beyond predicting the structures of individual proteins in isolation.
What does beyond individual proteins mean in this context? Because, as you said, proteins rarely work alone in the cell, right? They interact constantly.
You're absolutely right. That's the key insight driving this next step. AlphaFold 3 is designed to predict the structures of proteins in complex with other molecules.
Other molecules, like what?
This includes predicting how two or more proteins bind together protein complexes. It includes protein DNA or protein RNA interactions, which are absolutely fundamental for controlling gene expression and regulation. And crucially for medicine, it includes protein ligand binding how a protein interacts with a small molecule, like a potential drug.
So predicting exactly how a drug fits into its target protein.
Exactly. This is a huge step toward modeling the true complexity and context of biology, where proteins function as part of these larger interconnected molecular networks in machines. It's moving from understanding the individual parts to understanding how they assemble and interact.
Okay, so it's moving from predicting the shape of a single gear to predicting how the entire clockwork mechanism fits together, how the gears mesh and turn each other.
Precisely. That's a great analogy. And the results they reported for AlphaFold 3 are impressive. They claimed at least a 50% improvement in accuracy for predicting these kinds of interactions compared to previous specialized prediction methods, even doubling the accuracy in some important categories, like predicting how antibodies bind to their targets.
50% or more improvement. That's significant.
It is. It means the AI can now often figure out, for instance, how a drug molecule precisely nestles into an enzyme's active site, or how an antibody accurately latches onto a viral protein, with much higher reliability than ever before possible computationally. This capability to model molecular interactions at such high fidelity is, frankly, monumental for biology and medicine.
I can only imagine how this will accelerate drug discovery pipelines even further.
Yeah.
What does that look like in practice for, say, the pharmaceutical industry?
Well, a key expected advancement, and indeed the explicit goal of DeepMind's sister company, Isomorphic Labs, is the direct integration of these AI predictions into drug development pipelines, potentially accelerating them exponentially.
How?
With AlphaFold 3's ability to model protein-ligand interactions accurately, researchers can much more effectively screen potential drug candidates and even design entirely new drugs in silico, meaning via computer simulation, before needing to synthesize and test them physically in the lab.
So less trial and error in the lab, more targeted design on the computer first.
Exactly. Isomorphic Labs has already announced collaborations with major pharmaceutical companies to apply AlphaFold 3 to real-world drug design challenges. The hope is to dramatically shorten the time and reduce the cost needed to find promising drug candidates.
By doing what specifically?
By quickly identifying how potential drugs bind to disease-causing proteins, predicting their binding strength, and then computationally optimizing the drug molecule structure to improve that binding and its other properties. We can expect to see AI-driven platforms emerging that can suggest novel molecules specifically designed to block or stabilize proteins implicated in widespread diseases like cancers, Alzheimer's, autoimmune disorders, and antibiotic resistance. It could really revolutionize the toolkit for medicinal chemists.
This sounds like a future where we don't just understand proteins, but we can actively design and control biological processes at a molecular level. What about other types of biological molecules? Will this AI approach extend its reach beyond just proteins?
Absolutely. The success with proteins is definitely inspiring work on other crucial biomolecules. Researchers are actively working on AI models for predicting RNA structure. RNA is fascinating. It's not just a messenger. It plays complex roles in gene regulation, catalysis, and it's increasingly recognized as a significant drug target itself.
So AlphaFold-like approaches for RNA.
Yes. And interestingly, AlphaFold 3 already takes steps in this direction. It explicitly incorporates nucleic acids, DNA, and RNA into its prediction capabilities. It can model protein DNA and protein RNA complexes. It even considers things like post-translational modifications on proteins, which are critical chemical tags that cells add to proteins to regulate their function. All of this is moving us toward a more comprehensive digital biology model, aiming to simulate the key molecular players in the cell.
So it's not just predicting static snapshots anymore, but
Right.
potentially moving towards a more dynamic, almost living model of molecular activity within the cell.
That's certainly the long-term aspiration. Future AI systems might predict not just static structures, but also protein dynamics, how proteins change shape over time, how they flex and move, how they transition between different functional states. DeepMind also has a separate project called AlphaFold, for instance, which uses AI to predict which genetic mutations are likely to cause disease by assessing their probable impact on protein structure and function.
Predicting the impact of mutations.
Yeah.
That has huge implications for personalized medicine.
Huge. And another incredibly exciting direction, moving beyond just understanding, is protein design, creating entirely new proteins from scratch for specific purposes.
Like those plastic-eating enzymes.
Exactly. Imagine designing a novel enzyme to efficiently degrade a specific environmental pollutant, or engineering a therapeutic protein perfectly tailored to neutralize a particular virus, all designed and refined on a computer using an AlphaFold-like AI to predict its structure and stability before you even step into the lab to synthesize it. This would truly be a paradigm shift towards engineering biology with vastly reduced trial and error.
That's incredible, going from reading the blueprints of life to actively writing new ones. What about improving the core AlphaFold system itself? Are there still areas where accuracy or scale needs to be pushed further?
Oh, definitely. Developers certainly aim to improve accuracy further, especially for the really tricky edge cases. Some proteins remain challenging.
Like which ones?
For instance, proteins that are intrinsically disordered, meaning they don't adopt a single stable 3D shape until they bind to a partner molecule, or proteins that are highly flexible and dynamic, or extremely large multi-component molecular machines where predicting the arrangement of all the parts is still tough.
So how are they tackling those?
Work is ongoing to integrate different types of data. For example, combining AlphaFold predictions with sparse experimental data, maybe from lower resolution cryo-EM maps, or data about which parts of proteins are known to be close together, cross-linking data. The idea is to use experimental clues to guide or constrain the AI's predictions for these really tough cases. It's about synergistic combination of computation and experiment.
And what about the training data? AlphaFold was trained on the publicly available structures in the PDB. Is that resource now sort of tapped out for training these models?
That's a really interesting point. The publicly available PDB data was crucial for AlphaFold's initial success, but yes, in a sense, the models have now learned most of what's available in that public dataset. This means future significant improvements might need new or different kinds of data.
Such as?
Well, this leads to some interesting developments. For example, a consortium of major pharmaceutical companies announced plans in 2025 to pool their resources and train a new, potentially even more powerful AI model on thousands of proprietary protein structures solved internally within their companies, often of medically relevant targets that have never been released publicly.
Proprietary data. And that, I imagine, raises some questions about openness versus commercial advantage, potentially creating different tiers of access to this powerful technology.
It absolutely does. The tool trained on this private data might achieve higher accuracy on certain types of drug targets, but it almost certainly wouldn't be made openly available in the same way AlphaFold was. This points towards a potentially diverging landscape, a rich, evolving ecosystem of AlphaFold-inspired methods emerging, but with some potentially remaining behind corporate firewalls.
So we might see competing models, like Meta's ESM-Fold you mentioned.
Exactly. Meta AI developed ESM-Fold, which uses a different AI technique based on large language models, similar to those used for text like GPT. It's generally faster than AlphaFold 2, but slightly less accurate on average. So we'll likely see a diverse range of tools developing, optimized for different needs, speed versus accuracy, open versus proprietary. The future will involve tackling ever more complex interactions, deeper integration with drug discovery, and expanding this AI paradigm into a general platform for modeling all of life's intricate molecular machinery. If AlphaFold 2 gave us the individual parts list, AlphaFold 3 and beyond aim to show us exactly how those parts fit together, and crucially, how we might begin to engineer and control them.
This really does feel like the dawn of a new era in biology. So, taking all of this immense progress and future potential into account, what is the overarching impact of AlphaFold on biology and medicine? How is it truly ushering in this new age for understanding and hopefully curing disease?
While many leading scientists agree that AlphaFold's success isn't just an incremental improvement, it represents a fundamental leap in our ability to understand biological systems at their core molecular level. By quickly revealing the likely structures of virtually all known proteins, it's effectively given scientists a comprehensive lookup table, or a structural atlas, for the building blocks of life. It provides a structural lens through which to view almost all biological processes.
So it's like we suddenly have a detailed reference manual with pictures for almost everything that's happening chemically inside our cells.
Exactly. Before AlphaFold, our knowledge of protein structures was incredibly spotty. We had high-resolution pictures for some important proteins, sure, but vast portions of the proteome, the full set of proteins in humans, in pathogens, in plants, remained structurally uncharted territory. They were just sequences of letters in a database. Now, with high-quality structural models readily available for most of them, biologists can begin to interpret how these proteins actually work mechanistically.
And that allows them to do what specifically?
Researchers can now look at a protein implicated in a disease, see its predicted 3D shape, and immediately start forming hypotheses about how it functions, how it might interact with other molecules, or why a specific genetic mutation causes it to malfunction. Maybe the mutation disrupts a key binding site or prevents proper folding. This structural perspective is truly like suddenly gaining X-ray vision into the molecular world.
That's a powerful image. What has that X-ray vision already eliminated for us in terms of understanding disease?
It's already shedding light on the mechanisms underlying countless diseases: cancer, Alzheimer's, Parkinson's, diabetes, infectious diseases, rare genetic disorders, you name it. Scientists studying these conditions are using AlphaFold models every day to pinpoint where things might be going wrong at the molecular level, something that was incredibly difficult or impossible to do before for many proteins.
And beyond disease.
Yes. In evolutionary biology, comparing the predicted structures of related proteins across different species helps trace how proteins evolved their functions over millions of years. It can even help predict the functions of completely uncharacterized proteins based solely on their structural similarity to proteins whose functions are known. The sheer volume of structural data generated by AlphaFold is sparking new hypotheses and accelerating research across nearly every single area of the life sciences.
This direct molecular understanding must also significantly accelerate the development of new drugs and vaccines, right? You touched on structure-based design earlier.
Absolutely. This is one of the most direct and impactful applications. Knowing a protein's precise 3D structure vastly aids in rationally designing drug molecules that can bind to it effectively. Pharmaceutical researchers now routinely use AlphaFold models of their target proteins to guide the very first steps of drug discovery, identifying potential binding sites, understanding how existing drugs bind, or screening virtual libraries of compounds to see which ones might fit. It significantly shortens the often lengthy and expensive hit discovery phase.
So if a particular protein is known to drive tumor growth, they can visually inspect its AlphaFold model, find a vulnerable-looking pocket or groove on its surface, and then design a molecule specifically shaped to lock into that spot and shut the protein down, almost like designing a custom key.
Exactly. That's the essence of structure-based drug design, and AlphaFold makes it applicable to a vastly larger number of potential drug targets than ever before. We already discussed the benefits for neglected diseases. Similarly, in vaccine development, understanding the precise 3D structure of viral or bacterial proteins, the antigens, is absolutely key to designing vaccines that elicit a strong, stable, and protective immune response.
How does structure help with vaccines?
Because the immune system recognizes shapes, AlphaFold has helped researchers determine the most stable and relevant structures of antigens for potential next-generation vaccines against diseases like malaria or improved versions for COVID-19, allowing these vaccines to be designed more rationally and effectively. It truly empowers researchers across academia and industry to find potential cures much faster, and as we noted, it democratizes this capability, making sophisticated structural insights accessible even for fields that traditionally lacked the resources for extensive experimental structure determination.
And it's not just about targeting existing proteins with drugs, but you mentioned we're even moving towards designing entirely new proteins. That still sounds like something out of science fiction.
It's rapidly moving from science fiction to science fact. Understanding protein structures doesn't just help us target existing proteins, it fundamentally enables the design of entirely novel proteins with new functions. This field is often called protein engineering, or synthetic biology.
What can we do with engineered proteins?
Well, with these structural models as guides, scientists can rationally modify existing enzymes to make them work faster or operate under different conditions like higher temperatures, which is hugely beneficial for industrial processes and developing greener chemistry solutions. Or they can engineer completely new therapeutic proteins.
Like designing better antibodies.
Precisely. If we understand exactly how an antibody binds to a viral protein using an AlphaFold prediction, we can attempt to computationally redesign the antibody to bind even more tightly, or perhaps to evade viral mutations, potentially yielding a much better treatment or preventative therapy.
And the plasticating enzymes you mentioned, that's a perfect example of engineering biology for environmental benefit.
Exactly. In medicine, this capability could lead to custom-designed enzymes that specifically break apart disease-causing molecules within a patient's body, or protein-based drugs engineered for enhanced stability, better delivery, and higher specificity. In the long run, this opens the door to entirely new classes of therapies, think protein therapeutics or synthetic biology solutions, potentially tackling diseases that have proven intractable for traditional small molecule drugs or even current antibody therapies. It's a truly exciting frontier of biological engineering enabled by this deep structural understanding.
Really does feel like a new era for biology, maybe analogous to how the Human Genome Project provided that complete genetic blueprint decades ago, kicking off a new wave of discovery. The Nobel Committee's quick recognition seems to signal that computational predictions are now truly central to biological discovery.
I think many researchers share that sentiment. We often hear phrases like a new age for biology or that the molecular machines of life are no longer operating in the dark. With the basic protein structure prediction problem largely solved, scientists can now shift their focus and resources towards tackling higher order, more complex questions.
Like what?
Like, how do these predicted structures actually function dynamically within the incredibly crowded and complex environment of a living cell? How do they interact in vast, intricate networks to carry out biological processes? And crucially, how can we precisely manipulate these structures and networks for therapeutic benefit or biotechnological application? AlphaFold has essentially removed a massive bottleneck, accelerating progress across almost all of biochemistry and molecular biology. It allows us to ask much more sophisticated questions now, moving beyond simply identifying the parts list to understanding how the whole complex system works and how we might intervene.
But it's important to remember, it's not a perfect, magical crystal ball, right? There are still caveats, limitations, and maybe new frontiers that require human ingenuity and experimentation.
That's a crucial point to emphasize, absolutely. AlphaFold's predictions, while often remarkably accurate, are not perfect. They are predictions, hypotheses. There are definitely instances where AlphaFold's confidence score for a region is low, indicating uncertainty, or where the predicted structure turns out to be incorrect when compared to experimental data. This is especially true for certain classes of proteins.
Which ones are still tricky?
Proteins that are intrinsically disordered, meaning they lack a stable 3D shape on their own and only fold upon binding to a partner. Also, proteins that are highly flexible and adopt multiple conformations. And predicting the precise arrangement of subunits in very large dynamic multi-protein complexes remains a significant challenge. So experimental validation remains absolutely crucial.
Especially for medicine.
Especially for applications like drug development, where you absolutely need to be certain about the shape of your target. Critical predictions must be confirmed with rigorous lab measurements using techniques like crystallography, cryo-EM, or spectroscopy.
So it's still a very powerful tool, maybe the most powerful ever for this problem, but it's not a replacement for experimental biologists or the scientific method. It's more like an incredibly smart partner.
Precisely. It augments human expertise. It doesn't replace it. Moreover, it's worth remembering that AlphaFold doesn't fully explain how proteins fold so incredibly quickly and reliably in nature, or the detailed step-by-step folding pathway they take.
Right, it gives the answer, but not the process.
In a sense, yes. It gives us the answer, the final 3D structure, but it doesn't provide a clear first principles theoretical model of folding kinetics or the complex energy landscape that guides folding. The why behind that rapid, reliable folding process is still an active area of research. So some scientists would argue that the protein folding problem isn't completely solved in that deeper theoretical sense of understanding the underlying physics and chemistry of the folding process itself. Those fundamental questions remain important frontiers for biochemists and biophysicists to explore.
And predicting dynamics, interactions in the cell. Those are the next big hurdles.
Exactly. Challenges like predicting how protein structures change shape when they interact with other molecules,
Mhm.
dynamics, or how these huge multi-protein assemblies form correctly within the crowded environment of a living cell, often involving helper proteins called chaperones. These are still very much frontiers to conquer. AlphaFold cracked one massive problem, but biology always has more layers of complexity waiting.
This incredible breakthrough, AlphaFold, has clearly had a massive scientific impact. But it also inevitably raises bigger questions about how science is done, about ethics, and even about philosophy. What's been the general reception within the scientific community? You mentioned amazement.
The response has been overwhelmingly positive, yes, often bordering on astonishment. We heard scientists using terms like shocking, game-changing, a profound and structural advance. There was a palpable sense of relief and celebration that this problem, which generations of brilliant biologists had wrestled with, had finally yielded.
And the openness was key to that positive reception.
Absolutely. DeepMind's decision to openly share AlphaFold's source code and especially the massive database of predicted structures received widespread praise. It was seen as a major boost for collaboration, transparency, and accelerating science globally. As we discussed, millions of researchers have accessed and used these predictions, leading to a genuine explosion of new studies and hypotheses across biology.
That analogy of getting a GPS map after being lost at sea seems quite fitting.
It really does capture that feeling of suddenly having clarity where there was mostly fog before.
But I'm sure, alongside the excitement, there's also a healthy dose of scientific caution mixed in there. You mentioned earlier that prediction isn't validation.
Exactly. Scientists are inherently critical thinkers. While celebrating the advance, many quickly pointed out that just because AlphaFold predicts a structure with high confidence doesn't automatically mean it's the biologically relevant structure under all conditions. Experimental validation remains paramount, especially if you're going to base critical decisions like developing a drug on that structure.
And it still struggles with some types of proteins.
Yes, there's a clear recognition that some proteins, the intrinsically disordered ones, parts of very large, flexible complexes, are still challenging for AlphaFold, or its predictions are less reliable. So human expertise, careful interpretation, and complementary experimental techniques are still absolutely vital to get the full picture. The consensus really is that AlphaFold hasn't made human structural biologists obsolete. Far from it. It's an incredibly empowering tool that frees them from some of the more routine aspects of structure determination, allowing them to focus their skills on more complex biological problems, on understanding function, dynamics, and interactions. It solved one huge problem, and in doing so, it actually opened up many more interesting and deeper questions to pursue.
That's a really positive way to look at it, not replacement, but empowerment. Let's talk a bit more about the ethical dimensions in this open science aspect. DeepMind is a commercial entity, part of Google, yet they made this incredibly powerful tool and its predictions freely available. That feels like a significant choice in today's world.
It truly was a significant choice, and one that garnered enormous goodwill within the scientific community. DeepMind's decision to make AlphaFold's predictions freely available through the database was widely lauded as a democratizing move. As we've said, it allows researchers anywhere, including those in universities or countries with limited funding for expensive experimental setups, to gain crucial structural insights. It leveled the playing field considerably and set a very positive precedent for how AI-driven scientific breakthroughs can be shared for the common good.
But are there concerns that this might not always be the case with future AI tools, especially as the technology becomes even more powerful and commercially valuable? You mentioned the Pharma Consortium.
Yes, there are definitely ongoing discussions and some concerns about that. The plans by pharmaceutical firms to train new, potentially more powerful AI models on their vast proprietary data sets raise the possibility of a future bifurcation,
Bifurcation, meaning what?
meaning a split, where academic researchers might primarily use open models trained on public data, while industry possesses superior but closed-source AI tools trained on private data. Many in the scientific community argue strongly that keeping core scientific capabilities like this open is crucial for ensuring that progress is shared broadly, allowing smaller labs, academics, and researchers in developing countries to contribute to discovery, not just large corporations with exclusive tools. It's an ongoing tension between commercial incentives and the open advancement of collective scientific knowledge.
That's a critical issue for the future. And what about attribution? This ties into the ethics too. When an AI is so central to finding a solution, like predicting a structure, who gets the credit? Is the AI itself the team who built the AI, or the biologists who used the prediction to make a discovery?
That's another important ethical and practical consideration that the scientific community is still navigating. AlphaFold's predictions come from an AI trained on publicly generated data. So who truly deserves the credit when its prediction enables a new structural solution or biological insight? In practice, what we're seeing is that researchers consistently cite AlphaFold and the relevant DeepMind publications when its predictions contribute significantly to their study. And, as we know, the DeepMind team leaders, Hassabis and Jumper, received the Nobel Prize, reflecting a formal recognition by the community of the immense value of creating the tool itself. It certainly represents a shift in how we think about and attribute scientific discovery in the age of AI.
This leads us nicely into some really profound philosophical questions, especially about the very nature of scientific understanding. AlphaFold, as you explained, solved protein folding primarily as a prediction problem. It's incredibly accurate at predicting the final structure, but it doesn't necessarily provide an explicit step-by-step physical explanation for why a specific protein folds the way it does, so quickly and reliably. So the philosophical question becomes, is the problem truly solved if we don't fully understand the underlying why, the fundamental principles in a way humans can grasp? Or is a highly predictive solution sufficient in itself?
This is a deep and genuinely fascinating debate that AlphaFold has really brought to the forefront in biology. Traditionally, the goal of science was often seen as finding those underlying principles, developing elegant theories that explain phenomena. AlphaFold, however, is often described as somewhat of a black box. It learned patterns from vast amounts of data, and it produces incredibly accurate outputs. But the internal logic isn't always transparent or easily translatable into simple, intuitive physical rules.
So it gives us the what, but not necessarily the how or why in a traditional sense.
Exactly. For many scientists, particularly those focused on applications like drug discovery or understanding disease mechanisms, having the accurate structures, the what, is incredibly valuable and allows them to move forward, even without a complete theoretical understanding of the folding process itself. This highlights a potential new paradigm in science, where powerful AI tools can, in some cases, bypass the need for complete human mechanistic understanding by simply providing highly accurate, actionable predictions directly from data.
So it forces us to ask maybe a fundamental question about the purpose of science itself. Is the primary goal to generate useful answers and enable practical applications? Or is it also about satisfying that deep human curiosity for understanding the underlying reasons, the fundamental mechanisms driving the universe?
Exactly, and there's no single easy answer to that. It's likely both are important, but AlphaFold certainly pushes us to think about the balance and the changing nature of discovery. It also powerfully exemplifies AI not just as a tool, but as a potential partner in scientific discovery. It uncovered knowledge millions of protein structures that humans alone struggle to find for over half a century.
That has huge philosophical implications for the future of science, doesn't it?
It really does. If AI could crack protein folding, what other grand challenges in science, problems that have stumped us for decades, might yield to similar AI partnerships? Perhaps predicting complex ecological dynamics, designing novel materials with specific properties from scratch, maybe even understanding the intricacies of the human brain or the climate system. It certainly energizes fields like computational biology and beyond, demonstrating that these sophisticated, data-driven AI approaches can tackle incredibly tough scientific nuts and powerfully complement human intuition and traditional methods.
Looking even further ahead now, beyond the immediate scientific impact, what are the potential long-term implications of AlphaFold and technologies like it for humanity as a whole? How might it reshape our future?
Well, if the potential is realized, the long-term impact could be truly monumental for human health and well-being. By dramatically accelerating our understanding of disease at the molecular level and speeding up the development of new drugs and therapies, it could contribute significantly to tackling some of our biggest health challenges.
Like what specifically?
We could potentially see faster discovery of new antibiotics to combat the growing crisis of antimicrobial resistance. Maybe novel enzymes designed to clean up pollution or capture carbon dioxide more efficiently. Perhaps improved crops engineered for better nutrition or drought resistance based on a deeper understanding of plant proteins. And as you mentioned, personalized medicine could benefit immensely from accurately predicting how an individual's specific genetic variants affect their protein structures and functions, for example, using tools like alpha-missense to understand disease risk and guide tailored treatments.
These potential advances sound overwhelmingly positive. But with such powerful technology, the ability to understand and even design the molecules of life comes great responsibility, right? We need to ensure these tools are used ethically and for good.
Absolutely, that's paramount. While the vast majority of applications are clearly beneficial, we must approach these powerful capabilities responsibly. The ability to design or modify proteins, pushed to an extreme, does theoretically raise concerns about potential misuse, such as trying to design harmful biological
Is
agents.
that a realistic concern right now?
Currently, designing truly novel, functional proteins with malicious intent remains extremely complex and difficult, far beyond just predicting structure. The technical hurdles are still immense. However, it underscores the need for ongoing ethical discussions, responsible development practices, and appropriate oversight within the scientific community and by governments as the technology matures. But it's crucial to state that the potential benefits for medicine, agriculture, and the environment seem to vastly outweigh these hypothetical risks, especially with sensible safeguards in place.
And what about equitable access to the benefits? If this leads to new drugs or therapies, how do we ensure they reach everyone who needs them?
That is another critical ethical consideration. If AlphaFold and similar technologies lead to breakthroughs, new life-saving drugs, for instance, it's absolutely crucial that mechanisms are in place to ensure those benefits are accessible globally, not just to patients in wealthy countries. The initial open availability of AlphaFold's results was a fantastic start in democratizing the knowledge, but continued advocacy and innovative health policies will be needed to translate these scientific advances into genuinely equitable health outcomes for everyone around the world.
And finally, perhaps just an inspirational thought to end on. It feels like AlphaFolds is a powerful testament to human ingenuity, especially when different fields collaborate.
I think that's a wonderful way to frame it. AlphaFold's incredible leap forward wasn't just about AI, it required a brilliant blend of deep biological knowledge, decades of experimental data painstakingly collected by the community, and cutting-edge artificial intelligence techniques. It's a testament to the power of interdisciplinary innovation. It serves as a powerful reminder, perhaps, and a source of optimism, that humanity, when aided by increasingly intelligent machines and working collaboratively across disciplines, can indeed tackle problems that were previously deemed unsolvable. It really expands our sense of what might be possible in tackling other critical global challenges.
AlphaFold truly stands as a landmark achievement, sitting right at that exciting intersection of artificial intelligence and fundamental biology. It has unlocked the 3D structures of the proteins that underpin all life, accelerating science in ways that honestly were unimaginable just a few short years ago.
Yeah, it's really quite something.
From cracking a 50-year scientific puzzle to empowering researchers worldwide with an unprecedented atlas of protein shapes and now guiding entirely new medical breakthroughs, its unique deep learning approach and that stunning accuracy have truly set it apart.
And it really feels like this isn't just another incremental step forward, it's a fundamental shift. It feels like it's inaugurating a new era where AI systems are direct contributors to core scientific discovery, reshaping biology into a science that's much more predictive and maybe even more design-driven than ever before.
So this leaves us, and you, our listener, with a fascinating question to ponder. If AI, like AlphaFold, can provide incredibly accurate solutions to a problem as profoundly complex as protein folding, giving us the what, the structure, even without fully revealing the complete intuitive why, the detailed mechanism, what other seemingly unsolvable grand challenges, perhaps across any scientific field, might now be within reach through these powerful AI partnerships. And maybe more subtly, what responsibilities come with prioritizing these incredibly useful predictive solutions, potentially over striving for complete mechanistic understanding in our ongoing quest for knowledge?
It's a deep one to think about.
Definitely something to ponder as you go about your day.